Hi everyone, welcome to lesson 3.4, cellular transport. Make sure to keep up with your notes and vocabulary as we go through this lesson. So last lesson you saw these 8 characteristics of living things, and we know all life is made up of cells, right? So that was kind of the focus of our first few lessons for this unit. Today's we're talking about cell transport. Some of this will be a review from Unit 1, where we briefly went over plant transport and you had your introduction to cell transport then. But it's important for us to remember that cell transport is really important whenever we're talking about these concepts, number 5, 6, and 7. So living things obtain and use materials and energy, living things respond to their environment, and living things maintain homeostasis. So last class you also saw these two terms and we talked about organelle, cell organization, but today we're going to look at the cell boundaries in a little bit more depth. So the cell wall is a strong supporting layer around the cell membrane in some cells. Most prokaryotes and many eukaryotes, including plants and fungi, have cell walls, although animal cells do not have cell walls. Most cell walls are porous enough to allow water, oxygen, and carbon dioxide and other substances to pass through easily. Cell walls are outside of the cell membrane, and plant cell walls are mostly composed of cellulose, which is a polysaccharide. The cell membrane is a thin, flexible barrier that surrounds all cells, and it regulates what enters and leaves the cell. All cells contain a cell membrane, and most biological membranes are selectively permeable, which, if you remember, means that some substances can pass through and others cannot. So today, as we look at cellular transport, we're going to look at these structures in a little bit more depth. And if you remember, the cell membrane is also sometimes called the plasma membrane. Okay, so a lot of information on the slide, but we're going to kind of talk through this. So all cells contain cell membranes, and generally it's made of a double-layered sheet called a lipid bilayer. So if we look at this image, okay, keep in mind this portion right here, lipid bilayer, and bi, right, two. Okay, so it's kind of composed of these two sheets. So the lipid bilayer gives cell membranes a flexible structure that forms a strong barrier between the cell and its surroundings. So as we go through the lipid bilayer, we have to keep in mind properties of lipids, so kind of refresh your memory from Unit 2. When the lipids that make up cell membranes are mixed with water, their hydrophobic fatty acid tails, okay, here, cluster together, while their hydrophilic heads are attracted to water. And the result is the lipid bilayer. So if you can kind of zoom in, you notice that all the hydrophilic heads are exposed to the water outside or inside of the cell, but all the hydrophobic tails are kind of tucked away from the water. So the phospholipid is a type of molecule that is a main component of the cell membrane. So this right here is kind of essentially like a simplified structure of a phospholipid. And phospholipids are made up of two fatty acids, a phosphate group, and a glycerol molecule. Okay, next we have the fluid mosaic model. So as we're looking at this, this is essentially just the fluid mosaic model. Okay, embedded in the lipid bilayer of most cell membranes are protein molecules. Attached to many of these protein molecules are carbohydrates. Because the proteins embedded in the lipid bilayer can move around and float among the lipids, and because many different types of molecules make up the cell membrane, scientists describe this as a fluid mosaic. And remember, the cell membrane is selectively permeable, meaning it lets some things in and it keeps other things out. So let's review some of what we just saw, okay? So every cell has a cell membrane that regulates the movement of materials, and nearly all cell membranes are made up of a lipid bilayer, okay, in which proteins, carbohydrates are embedded. In animals, phospholipids, and again, this is just kind of a simplified drawing of a phospholipid, phospholipids are the main component of the lipid bilayer. So think and discuss this question, okay? Why do lipids self-assemble into a bilayer when exposed to water? So kind of pause here and think to yourself on this. So the hydrophobic end of the lipid molecules turn away from the water molecules, but the hydrophilic, okay, end of the lipid molecule is attracted to water, both inside and outside of the cell. With water on both sides, so remember outside of the cell, inside of the cell, okay, water outside of the cell, inside of the cell, the cytoplasm, mostly composed of water, okay. With water on both sides, a two-layer or bilayer system of lipid molecules forms, with the phobic portions, okay, within the membrane tucked away from the water. So those hydrophobic tails are kind of tucked away from the water on the inside. So first check for understanding, go ahead, pause here, and try these on your own. Okay, so number one, do animal cells have cell walls? That one was no. Number two, okay, do plant cells have cell membranes? Yes, all cells have cell membranes, also sometimes called a plasma membrane. Number three, do plant cells have cell walls? Yes, plants do have a cell wall composed of cellulose. Okay, number four, what is the cell membrane composed of? So this is a big, long answer. Okay, I put a little bit more detail in there, but that's just kind of to review that information from here. Okay, so make sure that you have this information, but the cell membrane is made up of a double-layered sheet called a lipid bilayer. Phospholipids were the main component of the lipid bilayer, and there's also things like proteins, carbohydrates embedded within that, and that's how we get that fluid mosaic model. Okay, so here's a quick review from Unit 1, and we're going to go over general cell transport. So we last talked about this on plant transport. So if you forgot that, perfectly fine, okay, but we are going to review this today. So there are two types of transport, passive and active. Passive transport is the movement of molecules across the membrane without energy, and active transport was the movement of molecules against a concentration difference using cellular energy. So why do we have to talk about this today? Okay, well, this is a very important concept. Every cell exists in a liquid environment, and one of the most important processes carried out by the cell membrane is to keep the cell's internal conditions relatively constant, or to maintain homeostasis. It does this by regulating the movement of molecules from one side of the membrane to the other. So what's this concentration difference or concentration gradient that we're going to mention today? Okay, so concentration gradient is a region of space over which the concentration of a substance changes. And substances will naturally move down their gradients from an area of high to an area of low concentration. And they're going to keep eventually moving until everything's evenly distributed or at equilibrium. Okay, so here is, okay, our first view of that. So obviously here we have a high concentration of molecules. Okay, you can think of that as like the solute molecules. Okay, as well as a low concentration on this side. So this concentration gradient just tells us kind of naturally what's going to happen is things want to move from a high concentration to a lower concentration until they reach equilibrium. So this arrow here is showing the direction of movement of the particles. Okay, so everything's going to continue moving down their gradient until we reach equilibrium. Okay, so that's just an important concept as we get to passive transport here. Okay, so remember, passive transport is that movement of molecules across the cell membrane without using cellular energy. Okay, so diffusion was a type of passive transport in which areas move from a high concentration to low concentration. And once the substance on both sides of a membrane is equal, okay, equilibrium is reached. So here's a good image of that as well. Okay, facilitated diffusion is also a type of passive transport, and the only difference between these two is facilitated diffusion uses some type of membrane protein, okay, or a protein channel, but it still does not require any additional use of a cell's energy. And then osmosis, okay, is the diffusion of water through a selectively permeable membrane. And remember, osmosis is a type of facilitated diffusion because water passes through an aquaporin, which is a special water channel, a special protein, okay, water channel. So again, osmosis is the diffusion of water. So this is going to look pretty familiar, but it's important that we review these concepts. So a solute was the substance dissolved in the solution, and the solvent was the substance that dissolved something, and water was known as the universal solvent. Okay, if you recall this experiment, if we have that barrier, okay, water, more sugar is here on that right-hand side, and then water is going to tend to move towards the higher concentration of solute, in this case that is sugar, essentially until the concentration is equal, okay? But in this case, that concentration is equal just because now you have evenly distributed water and sugar particles, even though you had a net movement of water to the right side, okay? So the general rule is always water is going to tend to move towards a higher concentration of solutes. So then we have our three types of solutions: hypertonic, hypotonic, and isotonic. So hyper, okay, remember, think hyper, think above. So like a hyperactive person is like above active. Hypo, okay, is below, and iso, think equal. So for hypertonic solution, when comparing solutions, the solution has a greater concentration of solute. A hypotonic solution, the solution has a less concentration of solute, and an isotonic solution, the concentration of solutes will be equal. Okay, so driven by differences in solute concentration, the net movement of water out or into a cell produces a force known as osmotic pressure. In plant cells, osmotic pressure can cause changes in the size of the central vacuole, which will shrink or swell as water moves into or out of the cell. Okay, so let's look at the rules of osmosis and a little bit closer. Okay, so water will move towards solutes. Okay, water will move towards the hypertonic solution, and water will move away from a hypotonic solution. So, as seen in the image, osmotic pressure can cause an animal cell in a hypertonic solution to shrink, and one in a hypotonic solution here to swell. Okay, because cells contain salt, sugars, proteins, and other dissolved molecules, they are almost always hypertonic to fresh water. Okay, so as a result, water tends to move quickly into a cell surrounded by fresh water, okay, like this, okay, causing it to swell. Eventually, the cell may burst like an overinflated balloon. But in plant cells, okay, osmotic pressure can cause changes in the size of the central vacuole, which we can see here. Okay, that's that space here. And then that change in the size in the central vacuole, and it can shrink or swell as water moves in or out. Okay, but the cell walls prevent the cell from expanding even under tremendous osmotic pressure. Okay, so notice how the plant cell kind of holds its shape in both types of solutions, while the animal cell really doesn't. So here's our first check for understanding. So pause here and try this out on your own. Okay, so beaker 1, 2, and 3. So let's look at this. Okay, so number 1, there is equal movement of water into and out of the cell. Okay, we can tell that by the arrows. And we're going to assume that's because the concentration of solute is equal, both outside, okay, of the cell and inside of the cell. This would be a cell in an isotonic solution, and the size of the cell does not change. Okay, number 2, there is a net movement of water out of the cell. Okay, the movement of the water out of the cell, this arrow is bigger. Okay, and that's because the solute concentration is higher outside of the cell in the solution. So this would be a cell in a hypertonic solution. The cell shrinks or gets smaller because the water is leaving the cell. Okay, number 3, there is a net movement of water into the cell. Okay, this arrow is bigger. And that is because the solute concentration is higher inside of the cell. So again, this would be like a cell in something like fresh water. Okay, so this cell would be in a hypotonic solution. The cell expands or gets larger because water moves into the cell. So now we're going to look at types of active transport, okay? And again, that's movement of molecules against a concentration difference using cellular energy. So we have two types, molecular and bulk transport. So molecular is whenever you have small molecules and ions which are carried across the membrane by proteins in the membrane that act like a pump. Many cells use protein pumps to move calcium, potassium, and sodium ions across cell membranes. So here's that example here. Okay, for bulk transport, okay, that's whenever we have large molecules and even solid clumps of material that can be transported by movements on the cell membrane known as bulk transport. So our two types are endo and exocytosis. Okay, so endocytosis is the type of active transport in which material is taken into the cell, okay, by means of infoldings, pockets, what have you, of the cell membrane. The two types of endocytosis are phagocytosis and pinocytosis. So phagocytosis is the cells taking up particles and packing it within a food vacuole, and pinocytosis is cells taking up liquid from the surrounding environment. Exocytosis, last one, is the type of active transport in which materials are moved out of the cell. So here is kind of a good summary of those three things, okay, in a little bit more detail, right? So here is energy from ATP is used to pump small molecules and ions across the cell membrane. Active transport, proteins change shape during the process, and they can bind substances on one side of the membrane and release them on the other. Okay, so again, think protein pumps for active transport. All right, our other two types, endo and exocytosis. Okay, within endocytosis, there are phago and pinocytosis. Okay, and that can be the membrane forms a pocket around a particle, then the pocket breaks loose from the outer portion of the cell and forms a vesicle within the cytoplasm. Okay, exocytosis is basically just the opposite of this. The membrane of a vesicle surrounds the cell material, fuses that with the cell membrane, and the contents are forced out of the cell. So here's another example. We see endocytosis, or the process of taking material into the cell. In this specific picture, this is a white blood cell seen here, and that's engulfing a damaged red blood cell by phagocytosis, which is one of those forms of endocytosis. The extensions or arms of the white blood cell are completely surrounding the red blood cell. And amoebas also use this method for taking in food. Engulfing material takes a considerable amount of energy and determination, just like when we eat way too much pizza in one sitting. Okay, so again, think that is active transport because it requires a lot of energy. So here is kind of our quick review, okay, because we did cover a lot. So remember, today our big idea was talking about these characteristics of living things. And every cell exists in a liquid environment, and that's one of the most important processes carried out by cell membranes is to keep the cell's internal conditions relatively constant, or to maintain homeostasis. It does this by regulating the movement of molecules from one side of the membrane to the other. So the two types, passive and active transport, and then within that we talked about these specific types. So diffusion, okay, type of passive transport, facilitated diffusion, and then osmosis was a type of facilitated diffusion. For active transport, we had molecular and bulk transport, and then within bulk transport, we had endocytosis and exocytosis. And then phagocytosis and pinocytosis are both types of endocytosis, or taking things in. So now at the very end of this video, I'm going to put in the example from Unit 1 if you need a refresher on overall passive and active transport. If you're feeling good about this information, the last part of this video is optional, but it's always good to review. So now that we've gone through the types of transport and you have the definitions down, I wanted to kind of provide a visual so that way you can tell the difference between the types. Also, I kind of like to tie in this fun like school analogy as we go through it. So for this part, you do not need to take notes. I'd rather you put your pen or pencil down and just kind of sit back and watch. So what's really cool about this is molecules are often going to behave kind of like people, specifically students, teachers, kind of that school environment. So for diffusion, okay, this first example, imagine this black box is my classroom. Okay, so we are students, here's the classroom. Okay, I pushed all of my students to the left side of the room in really tight, close quarters. So at this point, everybody's feeling extremely uncomfortable and they're like, Miss Hill, what are you doing? But I had told them all, okay, now you have to stand in this corner. Now, you as a student in the corner of the room, really close next to other students, what is your first thought? Your first thought is probably, I need to move away, I want my own space, I don't want to touch other people. So, requiring no brain energy, my students are going to want to move and disperse. And here they go. So, as students, again, diffusion is just moving from an area of high concentration to low concentration, or basically spreading out. It requires no energy and it is a form of passive transport. So by the end of it, after all my students have spread out, they're going to be nicely distributed around their classroom. And if I were to divide my classroom in half, you would notice there'd be an equal distribution on both sides, okay? So that is essentially diffusion. Area of high concentration to low concentration, no energy, no brain power is required. Next one, facilitated diffusion. So the only difference between diffusion and facilitated diffusion is a protein channel. So here I have my students, okay, my hallway, also known as a protein channel. Okay, all of my students have free reign of my classroom here and the hallway to the outside. Okay, so I say, students, today you can move and work wherever you want. So naturally, all my students that were packed in my classroom are going to want to move out and they're going to want to spread out. Again, this is still not requiring energy. Okay, they spread out naturally. They're going from a high concentration to a lower concentration. And even within my classroom, they are now spreading out. Oh, and this last guy chose there. Okay, so facilitated diffusion, high to low, no energy. The only difference is you are adding in a protein channel. Now osmosis. The only difference between osmosis and facilitated diffusion is your protein channel is now an aquaporin, okay, so that's the name of it, and the molecules, the students, are now water. So The only difference between facilitated diffusion and osmosis, again, is aquaporin is the protein channel and the students or the molecules are just water. So as they move, it's going to be the exact same thing that just happened, but I made the students blue just to show, again, it is water. And they spread out nice and even. And now all the students or water molecules are happy because they are balanced. So again, osmosis, passive transport, is a type of facilitated diffusion. Okay, so now whenever we add in solutes or dissolved substances in water, the rules of osmosis get a little bit different. Okay, so remember, this one that we just went over, we were dealing with no solutes. There was nothing in the cell itself or in the classroom itself. Water is just going to evenly distribute. As soon as you start adding in solutes, that game is going to change a little bit. So here, okay, new scenario. The hallway is still an aquaporin. Okay, now my classroom is a cell and it has water molecules in it. Okay, and then my students find out there is free candy outside. Okay, so there are my students. And there's the candy, all those pink boxes. Okay, so here are all my students. Within the cell, within my classroom, I always keep a little bit of candy. So, there's going to be some candy distributed. But the students find out there is free candy outside and there is a lot of it. Okay, so this side, outside, would represent a hypertonic solution. Remember, a hypertonic, hyper means above. So it has a higher concentration of solutes, in this case sugar or candy, outside of the cell here, okay, than inside of the cell. So there's more candy on this side than on this side. So what do all of my water molecules or students want to do? Well, naturally, they want to get some candy. So they're going to all start leaving my classroom, going into the hallway. They're going to find their candy. They're going to bond to it. And they leave really fast. Okay, so, and there's my one student left. My one student bonded to that one candy. All my other students left towards the hypertonic solution or the higher concentration of solutes. So what's going to happen is now that I only have one student in my classroom or one student in my cell, my classroom size is going to shrink. Okay, so the classroom or the cell will shrink now that it's lost all of its water or students to the candy rush. Okay, so my classroom's going to shrink. All the free candy outside, my students are gone, water molecules are gone. Okay, cell has shrunk. Okay, next scenario. This is an isotonic solution. So in this case, both my classroom and the outside of my classroom have equal amounts of candy or equal amounts of solutes, okay? So what's going to happen is my students here, they're pretty happy, they're balanced. What may happen is some students may kind of go back and forth, but you notice if one water molecule or one student leaves, another one's going to come back over. So there's going to be equal distribution of water moving back and forth, okay? So an isotonic solution, neither my classroom, the cell, or outside is going to expand or shrink. Water molecules are just simply going to go back and forth in a net movement of zero. Okay, so if one goes over here, one's going to come back. Okay, now last scenario. So you still have your hallway. Now my classroom has the water molecules and I refilled my candy jar. Okay, so I have like my normal amount of candy, and yes, I do keep candy in my classroom. Okay, so what's going to happen is there is limited candy or limited solute outside in the hallway. So here are all of my students. Here are my candy storage, which are secret, okay? And then here are all my students, previous students out in the hallway, okay? So my previous students, they know I keep candy in my classroom, or they know that I have candy in my cell. So what all of my previous students are gonna do is they are going to want to come into my class, and my students are gonna wanna claim that candy. So water molecules are gonna bond to it, but here comes all my previous students. They're coming in, they're claiming their spot, they are all getting the candy, and then a couple find out in the hallway they're going to stay there. So by the end of it, my classroom, all of my previous students, current students, they all rush into my classroom or into the cell to bond to candy or bond to the solute. So this over here, the hallway, would have been a hypotonic solution, okay, meaning that there was less concentrations of solutes or less candy in the hallway than what was in the cell or in my classroom. So all the water molecules rush to this side. Now it's going to happen to my classroom or cell. It's going to have to get way bigger to accommodate all of my new students, okay? So my cell is now this giant square here. It has expanded, so that way it can accommodate all of the new students. And then within the cell, these water molecules, these students attached to the candies, they would spread out as well. So kind of a fun thing to try at home. If you would like to try this, this is kind of seeing that in real time. If you have some gummy bears and water and or salt water, you can try this. So my first example, this would be a normal cell. This is a normal gummy bear. Okay, this was placed in an isotonic solution. You notice that it still looks the same. The net movement of water in and out of that gummy bear was equal. Okay, so that's going to be a normal cell. Now, if I put my cell or my gummy bear in distilled water, what happens to it? Well, it expands. All of the water that was surrounding this original gummy bear knew that there was sugar inside of it, so all of the water wants to go into the gummy bear, okay, and it's going to expand. So this would be a cell in a hypotonic solution, and oftentimes hypotonic solutions will be distilled water, because water is going to want to move into the cell. It's going to expand that cell. And then the last one, I know kind of at first look this actually looks larger than this. I just think the picture's distorted. But this last one was the gummy bear in salt water. Okay, and it was a very, very high concentration of salt in the water. All of the water that was in the gummy bear moved out of the gummy bear to be into the salt water. Okay, because the water was more attracted to the higher concentration of salt outside of the cell rather than inside of the cell. So water would move out of the cell and it would shrink. That would be a cell in a hypertonic solution. Now, last one, active transport. So there are many different types of active transport. For now, you just need to understand the basic definition. Okay, so here's the hallway, and now it is also a protein channel or a pump. Okay, so the only difference is now this protein channel has a pump. Okay, it has some energy involved with it. Okay, here's my outside area with less molecules and ions, and there's my classroom. Okay. So in my classroom, it's full. I have a lot of students. Okay, so full classroom or cell full of molecules, ions, or students, however you like to think of them. Okay, now here is the outside area, and obviously there's a little bit more room and less people. So naturally, all of the molecules or ions or students out here, they don't want to go back into a crowded classroom. Okay, that is against their concentration gradient. That's going to require some energy. Okay, but I am telling them, get back in class. You have to get back in class, get out of the hallway. So I have to apply some energy, okay? And then using my energy, I am going to make all of my students go against their concentration gradient or against what they want to do. And I'm going to have them go through that protein channel, pump them through, and I make them all go back into that crowded classroom. Now these molecules, again, they have to go against their concentration gradient. They have to be pushed to get back into this crowded classroom by a protein channel or pump. All right, so again, that is the end of this lesson. Here is that review slide one more time, okay? So thank you for watching, okay? Please feel free to reach out with any questions and make sure to finish up your notes and vocabulary.